Great, thank you very much. Thank you all for sticking around. My name is Michael Beyeler. I'm an assistant professor at UC Santa Barbara. And today I'd like to tell you about our recent work towards what I like to call a smart bionic eye, which is the idea of combining AI smarts with a visual neuroprosthesis. A lot of you may be familiar with the so-called bionic eye, which is really a microelectrode array that can be implanted directly in the retina or the visual cortex. This is not science fiction anymore. Two devices have already reached commercialization. One is called the Argus 2 device, developed by Second Sight in California, and the other one is the Alpha AMS from a company in Germany. Both of these are now defunct, unfortunately, but there are more devices in clinical trial as we speak, a handful or so, and there are dozens of devices in development. On top of that, there is a growing number of research groups that are conducting basic research to understand how these devices interact with the visual system and form perception. We are one of those, and we are lucky that we are able to collaborate with many different device manufacturers and other research groups so that we can push the field forward in general and not just focus on the particular technology. I wanted to show you a couple of examples of devices that are in development. These are designed for people who have had sight for most of their lives, but then perhaps lost it due to retinitis pigmentosa or macular degeneration, and thus might want their vision back. What I'm showing here are two examples of retinal approaches. Top left is the Prima device developed by Stanford and commercialized by Pixium Vision. Each cell there that you see is a photovoltaic cell, a low solar cell basically, doesn't need a wire. When you shine an infrared light on it, it will produce a small current that can activate the nearby neurons. On the bottom is a suprachoroidal approach where you don't even have to go into the retina. That is also in clinical trials right now. And then we have cortical approaches. Two examples shown here. Sorry, Dan, that I didn't add Neuralink, but we'll hear more about it in just a second. On top is the Orion, which is a surface electrode array. And then at the bottom, we have the ICVP, which has intracortical electrodes that get you closer to, let's say, layer 4. With all these, what all these devices have in common is a couple of components. You need, first of all, a brain. You need external camera or projector, something to get the visual input. This is typically mounted as a small miniature camera on a pair of glasses. And then you go frame by frame through a vision processing unit that converts this stimulus to an electrical pulse, or really a series of pulse trains that are usually sent wirelessly to the microelectrode array that sits, let's say, in visual cortex. So if you stimulate these different electrodes, you will elicit neural responses and ideally visual perception, which we can measure with psychophysics. And some work in my lab has focused on this forward transform, the question of how do you go from an applied stimulus to the predicted response. You could build a model bottom up, if you will, but you will have to account for like retinal remodeling, all sorts of nitty-gritty details. So instead, what we take is a top-down approach, where we try to black box the whole visual processing and just look at this transformation from stimulus to response. And so this black box we call the forward model. For a long time, people have assumed a relatively simple transform, basically a linear transform between stimulus and perception. The main assumption is that stimulating a single electrode elicits a small luminous dot, a phosphene. And if you stimulate a grid of electrodes, that should lead to a grid of phosphenes. All too often, we see simulation studies like this. On top, we see a Landolt C stimulus. At the bottom, we see the phosphenized version of that, where you basically tile the image. And then for each square, you kind of sum up the luminance, and that defines how bright the phosphene should be. So you see this pixel grid at the bottom, which can be better appreciated here if you have a more natural scene and maybe a larger number of electrodes. Now you get many small spots, okay? The third example here is being a bit fancier and taken into account cortical magnification. So we have near the fovea, we have smaller phosphines than in the periphery. And typically You could also assume that if you have a brighter pixel area, you might want to apply a higher current, and that could lead to a brighter phosphene. Unfortunately, we know that this is not how patients really see the world. We know this because we have done psychophysics with retinal implant users, where we stimulate a single electrode and we haven't drawn a touchscreen what they see. And so here are 3 examples of an Argus 1 and then 2 Argus 2 devices. And the shapes you see there are phosphene drawings averaged over 5 trials and centered over each electrode that produced it. So you should be able to appreciate that only a small fraction of them look like pixels, look like dots. We see simple geometric shapes ranging from triangles to arcs and wedges. And they seem to be different for every electrode. But since this is averaged over 5 trials, you know that it's pretty consistent across trials. It's just different from one electrode to the next. Okay. The second assumption that is violated is that phosphenes do not always add up linearly. That's the finding. My student Yu-Chin Ho recently looked at a large data set where first 2 electrodes were stimulated one after the other and then together simultaneous. And the phosphenes would sometimes add up linearly, but not all the time. And so we spent many years modeling this. This is work that started in Ione Fine's lab. And for the case of epiretinal implants, we were able to show that it comes down to 2 open parameters, rho and lambda, that describe how far the current will spread, either along a nerve fiber bundle, an axon bundle, or away from it. So by manipulating rho and lambda, you can produce either long streaks with a long lambda and a small rho, or you can produce a blob by having them sort of be the same. And if you move the electrode around on the retina, you can produce different shapes. On top of that, we know that phosphenes are a function of the stimulus parameters. So in a biphasic pulse train, where you could manipulate amplitude, frequency, and pulse duration, you will get different shapes out. For one, we know that if you increase amplitude, phosphenes tend to get larger, not necessarily brighter. But if you increase frequency, the phosphenes get brighter rather than larger. And then pulse duration also has a really intricate effect on the phosphene shape. By increasing pulse duration, you can potentially remove the streakiness. It's a bit hard to see on this slide. But long story short is that the phosphenes produced are repeatable to the extent that we can measure them with psychophysics and model them, so that we can start predicting for any combination of stimuli what the resulting artificial vision is. Of course, there are also temporal distortions, temporal dynamics. I don't have time to talk about this, but if you want to explore that, we built a Python library called Pulse to Percept, where you can take any of the available implants and combine them with any of the available models to predict what the artificial vision by a certain device would look like. I have some examples for you. Although we don't know for sure, here's one of our current best guesses as to what someone implanted with Argus 2 might see if they are in a busy subway scene. So this is the input image. We're somewhere in the subway. We see it's very, there's a lot of people walking around. There's a subway arriving in the back. So there's motion. There are signs a bit far away that we can't really read. And if you didn't listen to anything I just said and just downscale this video to the 6x10 pixel grid, which is the size of the Argus 2 array, you would just get a blurry version of this video. And you can still appreciate the motion. You lost all the detail, but it's clear that things are moving in front of you. However, if we now add this axonal stimulation that produces these distortions, we start seeing the vision change. And now it gets kind of a fisheye effect almost, right? It's kind of distorted, elongated more. And this can vary from patient to patient. With this model, we have now a way to model different patients and simulate their different artificial vision. And we can also start asking questions like, what would happen if we had more electrodes? So back to the 6x10 Argus 2 example, this is what it might look like. But now if we increase the number of electrodes and an equal spacing, we start to see more details in the scene. Here's an example, 48 × 80 electrodes. Now you can see that there are multiple people walking around. It's still not enough to read what's on the sign, right? But it might be good enough to avoid obstacles near you. But I think we can do better than that. Can we be smarter than that, right? So back to the schematic of how a stimulus leads to a response. What I've shown you is an example of this forward model. It's shown on the example of retinal implants, but we could build a similar model for cortical implants if we have the data. This forward model tells you for a given stimulus, what is the predicted response? But what you really want is the inverse of that, right? You want to know, how do I produce a desired target response? This forward model is typically highly nonlinear, so it's hard to invert, but I'm sorry, Ione, deep learning can do it. So we could put a deep net in line with a traditional equation-based model to learn an approximate inverse of that. That will then tell you how you need to stimulate the different electrodes in order to produce a desired percept. We would train this model by minimizing the reconstruction error, which is the error between the predicted and the desired response. Here's an example of some simulations that we've done. We call this the deep stimulus encoder. So that's exactly what I told you. It's a deep network that tries to invert the forward model. And if you want to publish this at a reputable computer science conference, you first have to show that this works on handwritten digits collected in the 90s. So we tried this on the MNIST data set. I'm showing you two lines of examples here. The DSC is the deep stimulus encoder, and the bottom is the naive strategy, which basically just takes pixel by pixel of the input stimulus and converts that to an amplitude on the electrode. Okay, so if your rho and lambdas are small, if the distortions are small, the naive and the deep approach look pretty much the same. The problem is that these values are rarely small in practice. Every patient seems to have a different value for rho and different value for lambda. So if you happen to come upon a patient with a long lambda, where they see everything very streaky, now the naive approach is starting to look worse and worse, whereas the top line, the DSE, still shows you the digits recognizably. And where the naive approach really has trouble is when rho is large. That means every phosphene you produce is really large and blobby, and everything blends together. What the deep stimulus encoder learns then is to avoid amplitude and focus more on frequency, such that you can preserve the detail and still produce the digits. But we can be smarter than that, because usually it's deep learning, so you need a lot of data. And usually this model assumes perfect knowledge of the patient-specific parameters. That includes rho and lambda. It also includes the implant location and orientation and a bunch of other things like how brightness scales with amplitude and so on. And typically you don't know that for a real patient. But what you can do is you can use a human in the loop approach. We can use Bayesian optimization to estimate the user-specific model that they have in their eye. The way this is done is by having patients compare two stimuli. We call this a dual. So let's see there's a target stimulus, and you have two ways to stimulate those. We call these Phi1 and Phi2. These are the two different models. You show them two stimuli, and all they have to do is to have to say which one is better. I prefer the first. And you repeat this for a couple of iterations, and the Bayesian algorithm will learn what the optimal parameters are of the model. So as the duals, as the game keeps on going, the duals get better and better. There's going to be one model that looks very much like the desired target, whereas the other one is the challenger that tries to figure out the parameters that are least known. And so in this plot here, I'm showing you how the error evolves over time. On the x-axis, we have the number of duals, and you can appreciate that after maybe 50, maybe 100 duals, you already have produced a percept that looks very close to your target digit, the number 4. A bunch of horizontal lines we have there. The naive encoding is up top. And then if you didn't have the human in the loop, that's the middle band. And so what this is saying that if we provided a program that a patient can take home, it would be a little boring, 100 duals or so, but what visual psychophysics is not boring? So they just do that at night. you could have this Bayesian approach estimate the user-specific model, and that might change over time. But we can keep doing that and get a much better result than with the naive encoding. We showed that this is very resilient to noise, and even if you have the model misspecified. Although I should point out this is all simulation, so we're very excited to try this now in real patients. Okay, but still we can be smarter than that. What I haven't told you yet is that you probably won't be able to turn on all the electrodes at the same time due to current safety limits and whatnot. And current implants don't provide a wide field of view. And that changes everything, because really how people use these devices is very different from how you would simulate them. It's a small field of view, tunnel vision, and they have to move their head around to scan the environment. And so they have to take in the visual scene piece by piece and then use deductive reasoning to figure out what's going on. So maybe first you look down, you see this bright flash that's kind of vertically oriented. Then you move your head up and now you get like fireworks. It's all glittery. And if you remember that you're standing outside in the park, maybe you can deduce that, oh, I'm looking at a tree. I first saw the trunk and now I see the leaves. So this is the kind of experience that patients report. It's very different from natural vision. It's almost like a new visual language that they have to learn. On top of that, visual outcomes are typically measured on behavioral tasks, such as a door finding task or walk along a line, wayfinding stuff that is far away from the single electrode controlled lab experiments that are usually done. But one thing we can use here, I think, are virtual environments. They offer an elegant solution to many of these problems. It's really hard to simulate on a screen, but with a virtual reality headset, we can embed our sighted users in this virtual experience of what a bionic eye might look like. So as they move their head around, as they move their eyes around, we can update the scene and we can recreate in VR the same experiments that people do in the clinic, and then compare behavioral performance of these virtual patients with the real patients. We can do this in a fairly sophisticated way. We can fit the setup to a specific patient, put the implant exactly where it is in the patient, use the exact same stimulation strategy, and then build the exact same virtual environment to test people on. So this allows us now to also try out new ways to stimulate, for example. We can do that rapidly and try different strategies and see if they help behavioral performance. So that leads me to the last point of my talk, which is really at the core of this is a question of how to best represent the visual scene. We often talk about these implants as a means to restore natural vision, where all current patient reports point to the opposite, that it's really not natural, and it's this new language they have to learn. So I'm asking, rather than aiming to one day restore natural vision, perhaps we should better focus about how to create practical and useful artificial vision now. And so what that will mean is focus less on vision restoration and focus more on practical use cases. For example, we can use computer vision to simplify the scene, highlight important objects, remove background clutter. There are many ideas out there in the literature. The challenge is not really to think up a new augmentation strategy. The challenge is to figure out what works for the patient. In which form factor should you provide this? Which cues are important to perform a certain task? And so that's really trying to connect this to the earlier session today. It's really more of a human-computer interaction problem. We can think of these devices as an accessibility aid, a tool set, right, that people can use. And so far, research has focused mostly on the technical aspects instead of the usability of these devices. What that means is that often they rely on a researcher's often cited perception of the end user's need. And only few studies really incorporate blind folk in the design and development of these devices. So our goal is to involve age-appropriate end users at all stages of the design process, use virtual patients for fast prototyping of new ideas, use real prosthesis patients for proof of concept experiments, and then iterate to find better ways of stimulating. And so that's really what I mean with the term Smart Bionic Eye. It's to combine these AI smarts with the patient-centered approach that works in everyday life. Sounds so easy, right? But that's really the super hard part. I think it's very unlikely that we'll find one simulation strategy that works everywhere and all the time. So it might be better just to focus on important tasks or contexts. If you have to do a wayfinding task, your needs, your visual needs are very different than if you're in conversation with people, okay? So with a blind person or a bionic eye user in the loop, we could figure out what are these visual cues that help you in this situation, and then come up with augmentation strategies that help you in these various contexts. We can use the virtual reality prototype to accelerate this loop. And then eventually, when we found something that works in VR, you can invest the time and money to bring it out to the real patient and see if it helps there. So long story short, how do I think we can get to a smart bionic eye? First, we need to use realistic phosphene simulations, because if we use these little dots, then our predictions may be misleading. Second, we need to move away from clinical tests to more real world tasks, ecological validity, right? And third, we have to think more about a patient-centered design where we involve end users at all stages of the design process. With that, I'd like to say thank you for your attention. Happy to take your questions.
We have time for a couple of questions.
Yeah, I'll start. Thank you. What happens with the eye movements in these patients?
Yeah. It depends a bit device to device. I would say most devices don't take them into account at all because the camera is fixed on the head. So instead of providing a gaze contingent scene, patients are instructed not to move their eyes at all. And that doesn't always work so well, yes. But certain devices are excluded from that, like the Prima device actually does incorporate eye movements. And I think that will greatly help people's understanding of the visual scene. Mm-hmm.
Hi, I saw your AR implementation of this at VSS and it was very nice. And so what I'd like to know is how much of what you've just talked about was in that simulation that was at VSS?
Actually, all of it, even more. What I haven't, well, I've only briefly mentioned that you cannot turn on all the electrodes at the same time, typically due to current limits. So what a lot of devices is doing is rastering the stimulus. You define a group of electrodes that you turn on, then the next one. So we had that in there. We had the temporal dynamics, mainly simulation of adaptation. So if you keep stimulating, right, the percept will fade out over time. And that's something that patients report seeing also. They're sometimes instructed to look into a dark corner if they can find one until the vision comes back. Yeah, does that answer your question? Thanks. Yeah.
Okay, why don't we save the rest for the end of the session? Thank you, Dr. Beyeler.